Hi, everyone, and welcome to this today's lecture in Worth Knowing. You have all seen the soup and the bread, I can see that, and there are some more soup for you to get. This lecture will be broadcasted, and you will be heard on the screen and film if you ask questions or stand in front of the cameras. We take all questions at the end after Sandra has finished her lecture, because I want to have the questions in the microphone so we get it on the streaming and on the film. This lecture series is an opportunity for us to discuss our great research and environmental monitoring and assessment, and I hope you will have some questions after the seminar. But now I would like to introduce Sandra Jämtgård that will talk about leftovers or a buffet. Welcome.
Thank you so much, Pernilla. I would like to first thank the organizers for giving me the opportunity to talk today, and I would like to thank all of you here in the room for taking your time to come here and listen to me today. So the research that I'm doing together with colleagues and collaborators focus on what roots experience in forest soils. You might think that we already know this, and I would say yes, to some extent we do, but at the same time, we actually don't. And the reason for this is that what the root is experiencing in the soil might not actually be what we are analyzing in our labs. And this is because during sampling and sampling processing, some of these compounds are, composition is actually altered through the processing. So what the root is actually experiencing in the soil might not be what we analyze when we get to the lab. So I want you to imagine you are a root growing in the soil, because the technique that we are working with is sampling the soil in the same scale as a plant root, in a very low intrusive way, in intact soil. And we're using a small membrane to do this that has the same scale as a plant root. And you can see it here on this image. It's a bit blank maybe, but you can see it there. And this photo is taken by a great friend and a colleague of mine, Richard Bracken. And this is from sugarcane soil in Australia. And you can see that this membrane is the same scale as a sugarcane root. So this novel soil sampling technique is called microdialysis. And it was developed in medical sciences for sampling, for example, neurotransmitters in brains. What is similar between a brain and soil, believe it or not, is this matter that when you take a sample, you're actually altering the chemical composition. And if you really want to know what the composition is, by using this technique, we are sidestepping these artifacts that are changing the chemical composition. So we have discovered that by using this technique in soil science and sampling organic nitrogen, we've seen this effect of this technique. So this technique, the application in soil, was initiated in the group that I'm working in here in Umeå. And by a former postdoc, Erik Inselbacher, he applied this in soil. And you can see this for organic nitrogen, which is nitrogen in the form of amino acids and proteins, for example, which is nitrogen which is attached to hydrocarbons. So when applying this technique in boreal forest soil around here in Umeå, he discovered that it was actually amino acids that was the dominant nitrogen form in the soil. And this was a bit contradictory to previous findings when we thought that ammonia is actually what the root is sensing the most in the soil. Similar findings was found by our colleagues in Australia in sugarcane soil, Richard Bracken and Susanne Schmidt, applying the same technique in unfertilized sugarcane soils. They found the same difference, that they actually saw a dominance of amino acids. And this is particularly interesting since the mechanisms for plants to take up these compounds are known, and that plants can actually use these compounds for growth, which has been one of the main focuses of Torgny Näsholm's group's research. So how is this possible? What's special with this technique? So what's special with this technique is, well, in one way, it's not that complicated. It is a semipermeable membrane, and it's a syringe pump, and that's what we're using for this. The thing with this semipermeable membrane and the probe that it's attached to is that it has the same scale as a plant root. So the ones we're using are about 1 to 3 centimeters long, and it has a diameter of 0.5 millimeter. And it can be inserted carefully, as you can see here on the images, into the soil when it's intact. So it's a very low disturbance. And the way that it's sampling the soil is that we are inducing diffusion. And we're doing this that when this probe is inserted into the soil, we pump ultra-pure water through this membrane. So this ultra-pure water has like 0 concentration of the compounds that we want to sample. And then on the outside in the soil, we have a high concentration of these compounds. And then the compounds travel, or they are transported, they move into the probe because it wants to go from high to low concentration. So it follows the concentration gradient, and it's then pumped out, and we can collect the sample in the soil. And this is a similar manner as a plant root would actually be how the compounds would be diffusing against a plant root. So when a plant root is taking up nutrients, there is also a decreased concentration, and it's a concentration gradient which is created, and these compounds are diffusing to the plant root. So the reason why we see this difference between traditional soil extract and by using this technique is the low intrusiveness that we can do in situ sampling with a very low disturbance. The other reason for this is probably because of the semipermeable membrane that is used that separates the samples from microbes and enzymes instantaneously when we take the sample. So a semipermeable membrane works in that way that it allows only compounds of a certain size to come into the sample. And in this case, it leaves microbes and enzymes on the outside, which don't go into the sample that we collect. And therefore, it represents more, they can't affect the composition during the transportation to the lab and when we are analyzing it. And just for you to get a view of what it actually looks like, this is an electron microscopy image of the actual probe membrane. So this is what it looks like. So we have done quite many studies with this technique on amino acids and nitrogen, since that has been the core of the research that we have been doing in the group. But of course, there are other compounds that we can sample with this technique. But of course, if you only analyze one type of compounds, you won't know about the others, but they might actually still be there in the sample. So therefore, we thought that it would be cool to actually look what else we're sampling with this technique. And there are other compounds which are prone to very fast degradation in soil. So the degradation or the turnover of amino acids in soil is like from minutes to hours. So it's turned over very quickly. Sugars are another compound which is turned over very quickly. So we thought maybe we should look at other types of compounds. So we did this project which was carried out by a former postdoc of mine, Värt, where we did metabolomic screening of the samples that she collected in a pine forest at Åheden, which is inland from here in Umeå. And she could, by this metabolomic screen, identify 50 compounds in these samples. But in total, she actually detected nearly 300 compounds. So 50 is a lot, but 300 is, of course, even more. And the reason for this similarity between what we could actually identify is because there is great potential in metabolomics, but there are also limitations. So the way the compounds are identified by metabolomics is based on libraries. And these libraries are mainly developed based on human samples, like blood samples, and plant samples. Soil is not that well investigated nowadays, so not yet. So what this indicates is that we have great future potential in finding even more compounds that we can sample with this technique. But out of this, nevertheless, 50 is quite a lot, and I was very happy for that. So Värt continued to do targeted analysis, which means that she was quantifying these 50 compounds, how much of them that we could actually sample. And it wasn't very surprising that she found amino acids. We knew that already. And organic acid wasn't a surprise either, because in other studies before we know that there are organic acids in these soils, but we hadn't used it for this technique for this particular case. So that was new. What was also a bit striking with this results was that we had the dominant organic acid was lactic acid, and that hasn't been found. It's been found that it occurs in soil, but not that it was dominating to the same extent as we found. Another thing that we found, which was slightly different to the previous studies on these type of compounds, was the proportion of sugars. We found less sugars compared to the literature, compared to like the proportion compared to amino acids and organic acids. Normally, there are more sugars in other studies. And we think that the reason for this is the low intrusiveness of this technique. Because when you do a traditional soil extract, you might actually be breaking some of the microbial cells, and there it might actually be sugars coming into solution. So we think that might be the reason why we saw less sugars. We also saw a larger diversity of sugars compared to other studies with this technique, which I think is very interesting. I will not go into where these comes from and their origin or nothing, because I will talk about the technique today, but I think this shows great potential, and I think it's very interesting to see what we can do with this technique. But the way that this technique is sampling the soil, that it's sampling flux rates rather than giving a snapshot of concentrations and the scale of it, that means that we can actually kind of say that it's mimicking a root. But in our way to simulate a root even more, there are other root functions which are also influencing nutrient availability to the plant root. So we are very curious about finding out what more we can do with this technique. So here you can see some of the root functions that have been or that we could potentially in the future simulate by microdialysis. So diffusion I've already touched upon. I will not go into all of these root functions today, but I will focus on mass flow and exudation, and I will touch a little bit on microbial interactions as well. Another thing which is influencing the availability of nutrients to the root is, of course, the transporters, which are responsible for the actual uptake of these compounds. What we don't have with this technique is that it's not selective. So the uptake transporters, they can be selective. So it's one transporter which is only taking up this type of compound and another one which is taking up that type of compound. So of course that we can't really at this stage do with this. But also another thing that transporters have is that they have different affinity. So they might have different efficiency of taking up different types of compounds. And that we can simulate with this technique. Another thing that plant roots have in order to get access of nutrients, that's growth. So they growth in the direction and finding nutrients on its way through the soil. And another thing is that different types of roots, different sizes and different developmental stages have different properties, and therefore they might experience different types of nutrients. But going back to mass flow and exudation, I will start with mass flow. Because mass flow is another way of roots to get access of nutrients. So diffusion, that's when the nutrients are going transported to the root surface along a concentration gradient. But the plant is also transpiring. And when it's transpiring, this gives a negative pressure in the root, which means that the root is almost like a straw when you're drinking a drink. So it actually makes body of water move to the root. And this makes also the nutrients that are dissolved in this water reach the root. So this is actually a benefit that plants have that microbes don't have. And with this technique, we can actually do direct measurements of what's potentially available to the root via mass flow. I will not go into details exactly how we do this practically, just trust me, we can do this. And if you have any questions about this, I can explain to you later. But when we did this, this was a PhD project that Olusegun Oyewole carried out when he was here some years ago. So he and I went out to the forest and we applied this technique in a pretty poor pine forest, halfway between Umeå and Vindeln, and a very rich spruce forest. For some of you might be familiar with Kulbäcksliden. And what we could see is that the nitrogen delivered to the probe via diffusion, that there weren't a difference between these sites, even though we clearly can see that there is a difference in growth at these sites. But when we measured mass flow, the nitrogen that would be available via mass flow potentially, we saw a huge increase in nitrogen availability. So this means that by mass flow, the plant can definitely increase the availability of nitrogen. Another thing which is influencing nutrient availability to the plant root is root exudation. So root exudation is something that the plant is believed to do actively or passively. It's a bit debated, but that they are releasing carbon compounds, or different types of compounds, in order to get more nutrients, like chemical-wise, or by stimulating microbial growth. And this can be, of course, symbiosis, but it could also be competitive. What is debated when it comes to root exudation is the composition of root exudates. And this review came out last year, pretty much concluding that it's mission impossible to actually sample what root exudates are composed of. And the reason for this is that the way we are studying this today is either doing it under axenic conditions. So that means like sterile with only plant roots available, normally in hydroponics, or that it's carried out in the soil or in the field where microbes are present. And even though we try to use very fine methods for doing this, it's still a bit tricky to distinguish what is coming from the microbes and what is coming from the root. Whereas in the lab, when we do this under axenic conditions, well, then it's difficult to say what the interaction with microbes, how they would actually influence this exudation. So we kind of lost in between there. Of course, we know some things about these things, but still exactly the precision of this, they're questioning in this review. But I would like to say, though, that with microdialysis, we are able to twist this in a slightly different way. So if we just want to look at the effect of root exudation, we can actually use this technique to simulate root exudation. So if we don't focus on the actual composition, but the fact that the root is delivering carbon to the soil by doing this. So a couple of years ago, Richard Bracken and Scott Buckley came over here from Australia, and we did some joint experiments. And then what they did was that instead of pumping ultra-pure water through the system, they were pumping what we say as exudates. So a known composition of carbon compounds. And by doing that, by pumping that through the system, we're simulating root exudates. So these compounds are then released from the probe. Simultaneously, we can sample this and we can look at how this is affecting nitrogen delivery to the probe, or what we believe is then mimicking a root. So the idea behind this is that plants are releasing this carbon to stimulate microbial growth, and microbes will then help the root to mine the soil for nitrogen and delivering it to the root. So that's what we expected. And as always in science, it didn't turn out the way we expected. So we actually saw the opposite. So we actually saw what's called immobilization. So when we pumped out these exudates, we saw a decrease in nitrogen delivered to the probe. So this is a bit where we are at the moment. We are trying to develop this in other directions. So this is a very conceptual way of looking at this. So we want to conceptually show that this is possible, but then of course you can do many, many studies looking into how different types of compositions is affecting this and so on. But right now we want to understand why this is happening. So Mark Swain, who's just finished his postdoc, have been working on that we want to see who is actually responsible for this immobilization. So on this electron microscopy images of the microdialysis probe, you can see that we are stimulating microbial growth. So what he did is to sequence the DNA from these probes for us to identify who is actually there on these probes. And what we are working on right now is that we want also to understand the mechanisms behind this. So Scott Buckley, who's now doing his postdoc with me, during his PhD he was developing the microdialysis technique from sampling soil enzymes. So soil enzymes are then responsible for these reactions, to this response. So we want to apply this and look into what is actually going on when we see this immobilization. And I'm very excited about this, and I really hope that we can use this in the future for looking into nitrogen availability, nitrogen use efficiency, and many more things. And then I guess I should go back to my title, which was a buffet or leftover, and I will simply conclude that it seems like there is a buffet out there, which I think is very exciting to know more about. And I think there is much more to know about this buffet. And I would like to thank everyone who's contributed to this. Several persons here in Umeå, in Australia, in Vienna and in Canada, and of course the platforms that we use and the funding bodies. Thank you.
So we have time for a couple of questions. And then I think Sandra might be able to stay here after the questions, and you can come confront her with more questions. But now, questions? Yes.
Very nice talk. Thank you very much.
Thank you.
I was just wondering, can you address the drought response of the roots to drought? How they will, with this technique that you're using, like maybe by changing the amount of water
you mean like
you
when
have
it comes
inside
to
the matrix?
You mean how it affects the nutrient availability?
Yeah, and if your technique could be used to address drought response.
Yeah, I guess to a certain extent. What might be limiting is because we are pumping water through the system, so there is a threshold for how low in water holding capacity we can go when we don't get a sample where the technique is not working. But except for that, yeah, we can measure at different amount of water availability.
Okay.
Yes, some more questions? Well, I have a question. So how can this be used out in the real world? Is it?
That's a good question. We are working at a very small scale, of course. But what we're hoping is that we can then later scale it up to understand, like, if you apply a fertilizer, how is that influencing the availability of nitrogen availability for the plant? But of course, like, the weakness with this technique is, of course, that it is a small scale. So if you want to go out and that you need this equipment in order to go out and do it, I guess the company selling this probably would like that everyone would have one of these in their backyard. But I think soil extracts and soil samples, that has a purpose as well for certain things, and this has a purpose for other things, for understanding the mechanisms behind it.
Very exciting sort of methodology and the possibility to approach to see things we couldn't see before. I was just thinking how I mean, most roads they are sort of covered with the fungal hyphae, and how, I mean, it doesn't affect availability, but how does it affect sort of, how do you handle that interpretation of what they take up? Because you have the different fungi, have different properties and different probably different ways of affinities and abilities to take up different types of compounds.
Yeah.
So how do you, if you could reason a little bit around that, please.
Yeah. So I'm hoping that we can look into that and understanding how they are working and see if what we are sampling is affected by that, if we can kind of align that. I'm very curious about doing that and how it's affecting the diversity of fungus as well, like when you apply fertilizer and so on. The problem we can have methodologically is that if the whole probe is covered, that is actually influencing what we are recovering, and that we are looking into at the moment. And I guess by using internal standards and so on, we can have an idea of how much is affected by the fungi and how much is affecting by what is transported from the outside, kind of. I'm hoping, but that's where we are at the moment, and I'm hoping that we can, figure that out.
Some more questions? No. Then we thank Sandra for an excellent talk, and she will stay here and answer more questions if you want to confront her. And before we end this, I would like you to see the schedule for the rest of Worth Knowing this term. Take a look at that also before you leave this room. But thank you, Sandra.
Thank you.